Hi, my name is Andrew Briley and I'm going to describe the figure in Chapter 7's current focus box on microlayers. So a microlayer is a layer in the ocean that is very thin, and they form for a number of physical reasons. And one of the reasons that was put forward in 2009 in a really nice paper in Science by Durham and Co. Was called gyrotactic trapping. And I'm going to explain how this gyrotactic trapping occurs. Durham and Co. worked on Chlamydomonas nivalis, which is a single-celled green algae about 10 micrometers in diameter. And this cell has a couple of flagella that enable it to swim. And these these plants swim upwards to enable them to remain in the illuminated upper parts of the water column. They're photosynthetic. Now, the structure of these organisms is such that their centre of buoyancy, the centre of the organism where buoyancy is focused, is different from the centre of mass of the organism. So in panel A of the diagram here, the centre of mass is shown by the red dot, and it's towards the rear of the organism, whereas the centre of buoyancy is at the middle of the circle. The organisms are portrayed as circles. They'll never be ideally circular. And panel B shows the behaviour of these organisms in different current shears. So what do we mean by shear? So shear is the rate of change of velocity with depth. So depth is shown by the y-axis in panel B. Depth is typically given the z dimension. And what we can see is in the upper part of this panel, the current is strong and consistent to the left, whereas at the bottom, the current is strong and consistent to the right. In the middle pane, there is a transition between strong flow to the left and strong flow to the right. So the shear in this zone is really quite strong. So the goldish brownish lettering against the slope that's plotted on the rate of change of current flow is the gradient or the shear rate. And when the rate of change of shear is high, where within a short vertical extent you're going from a leftward flow to a rightward flow, this leads the individual cells to tumble. So let's think about organisms swimming in the top to start with. Because the entirety of an individual organism is subject to more or less constant flow, constant to the left at the top, the organisms are able to swim in a constant direction. And the same is the case at the bottom. However, in the middle panel, where current flow changes really rapidly with depth, this conspires to tumble the individual organisms. And this means that rather than progressing in a uniform upward direction, where the distribution of cells would remain constant, organisms that swim into this high shear zone or sink into this high shear zone if they're coming from above tumble. And because they're tumbling, they don't escape from this zone, and so they accumulate. And so one of the things that Durham and Co. have suggested is that a mechanism by which thin layers can form is the trapping of cells in this high shear zone. And one of the manifestations they were able to demonstrate in the laboratory was that by manipulating shear rates, they could provoke thin layers to form. And thin layers are important in the open ocean, in reality in the open ocean, because they're a concentration of cells, and concentrations of cells are more productive for foragers than our dispersed cells.